your reaction in the chat box. So I might probably need an audio response if there is a need. Are we agreeing with that? Because I want to go to full screen now, so I won't be able to see the chart. So I will need an audio response whenever there is a need for that. Okay, thank you. Oh, okay, that's fine. That's that's fine. All right. So can we see the full screen, please? All right, thank you. So I just need one or two people to tell me their understanding of when you hear the word. Of course, we know what gene is, so that we don't start with. I believe one of our facilitators must have taken us molecular biology by telling us what is DNA, what is gene. So I believe they've done that already. So I want us to just move on so that we don't need to repeat most of the things we've been taught. So, but if you hear the word gene editing, what comes to your mind? I just need one or two people to say something.
Yes, sir.
If you want to say
Good evening.
something, you can just use my ear. Abdullah, yeah, you have the floor.
Yes, ma'am. Thank you, ma'am. And I thank our lecturer. Thank you, sir. So, as for gene editing, what comes to my mind is this general controversy around genetically modified organism, like the use of the editing of gene, like the modification of it, so as to produce better ones. So, yes, sir.
Oh, thank you. Sorry, I can't see your name, so but thank you. All right, that's a thoughtful and please, one thing I will appreciate is I love the fact the way it was sincere. So there is no right, there is no wrong. If you know it already, there is no need for you to be here. So be open to say whatever you know. That is why we are here. If you're correct, we'll tell you that you're correct. If you're wrong, then we'll correct the error. So please feel free. Thank you so much again for being sincere with your answers. All right, so can I have a second person before we go ahead?
Uh, Thank you so much, ma. Thank you, sir. Yeah, I think gene editing, like this, this which allows experts to modify the DNA of an organism.
Okay. All right. Thank you. All right. Thank you, guys. So it means we're on the same page. So I'm confident that I should be able to take us further then. All right. Thank you for the two speakers. One said what comes to his mind first is genetically modified organism (GMO), and the other said how the experts use it to modify the DNA. In all the way, you guys are correct in respect to different contexts. So when we talk about genetically modified organism, of course, the gene editing is not the only tool that is being used in genetically modified organism. So when you hear gene editing, it shouldn't be the genetically modified organism that should come to your mind because it is not only that. Even before the invention of gene editing, there has been genetically modified organism. Before gene editing comes to, you know, take everything up, genetically modified organism has been existing. So I wouldn't want you to have that picture of whenever you hear the word gene editing, what comes to your mind is genetically modified organism. Okay. So the next person that said used to alter the DNA sequence or alter the DNA, you are right. So now we're just going to go through it and get familiar with what the gene editing is. So if it is possible, maybe I will share the slide with the convener. However, if you can take note, take. If you can screenshot whatever you want to do, just make sure you're listening and you're following. So like I have on the screen now, genome editing, you can call them gene editing, genome editing, of course, still is still in context. Okay. So the genome editing, also known as genome engineering or gene editing, it involves precisely adding removing, altering, or substituting DNA sequence within the genome of a living organism. Why I try to make this decision, why I try to make this definition in this such a case is that there are some words that should be included when you're trying to describe genome editing. There are some words that are needed. There are some keywords that makes your definition, would I say, true, or that makes your definition make sense. Because the gene editing is not just for you to add alone. It's not just for it. It does different things depending on how you design your experiment, which we are going to be seeing later in the class. So it can add, maybe you want to introduce a mutation or you want to introduce a sequence in a gene, in the DNA. So you can use the genome editing to introduce that particular sequence. So that is adding. In other words, we call it insertion. You're trying to introduce a sequence. You're trying to probably create a mutation or probably, yeah, just let me say introduce a mutation. Because if the DNA is normal before, you adding something means you're trying to create a mutation because that is a change in the genetic material. So you can as well use it to remove. So maybe you don't want to add, you can use it to remove if there is any particular mutation in the gene, maybe due to point mutation, frame shift mutation, chromosomal rearrangement, whatever mutation that is going on in the gene. You can use genome editing as well to remove them. Please, are we together or am I too fast? If you don't, definitely. All right. So I'm trying to explain the context of the keywords here. So that is why I'm trying to break it down one by one. Okay. So we've talked about adding, we've talked about removing, which you can use if there is any mutation. Take for example a cancer now. Of course, we all know that cancer is as a result of mutation, probably due to point mutation, gene amplification, or whatever mutation that happens inside the gene. So you can use the genome editing to remove that part, that mutated sequence or that mutated region. So you can use to alter the sequence. You can use to, just like the word I've explained before, you can use to substitute. Maybe when you have a guanine in the gene, when you do your sequencing, you have a guanine. You want to, maybe the guanine is a mutation. What ought to be there is no guanine. It ought to be cytosine. Because when you have a single nucleotide polymorphism, when you have a single nucleotide variant, it can cause whatever to the organism. So in that case, you want to use your genome editing to switch your guanine into cytosine or your guanine into adenine, depending on what ought to be there, comparing with your database. So that is substituting the DNA sequence. So these are the things that genome editing can do for you. So it can add, it can remove, it can alter, it can substitute the gene. So, and the second point I have here is that unlike the traditional genetic engineering method that can just only insert genetic material randomly. That is why I said it before that before the discovery of the gene editing, all these things has been going on. But it's just that most of the time they are not specific; they are just randomly done. So these are more or less like the traditional genetic engineering. All this your crossbreeding, these especially those people in or would I say the agricultural parts. They've been doing crossbreeding, they've been doing all of those stuff before this genome editing came up. So the gene editing allows for a targeted modification instead of just randomly crossbreeding, instead of just randomly joining sequence together. It allows for a targeted modification. Oh, this is what I want to introduce. This is what I want to remove. This is what at a specific site. So if the genome editing makes it specific, it makes it sensitive, it makes it targeted. So it gives you that upper hand compared to the traditional genetic engineering. So now we have different types of this. I'm on the next slide now. Can we see that? So the mechanism can also be called homoendonuclease. So maybe in you're reading some journals, you can see homoendonuclease Some journals can call it meganuclease. It's still the same thing. So they are natural enzyme that cut the DNA precisely. But one of the things that is associated with them is that they are hard to design. And as well, of course, there is a limiting use of them. And it has, I mean, I would say that is one of the first genome editing technology. So it is because it is hard to design, which led to the, would I say, people not using it. Because maybe by tomorrow we get to see the design part. Because one thing that you need to do in your genome editing experiment is the computational aspect. How do you design your experiment, which we're going to be seeing tomorrow? So the next technology, of course, we're still going to take them one by one to just get ourselves kind of familiar with them. So the next one is the zinc finger nucleus. The zinc finger nucleus, which is called ZFN. They are another endonuclease. When we say endonuclease, it means an enzyme that has the ability to cleave. They have the ability to cleave. That is what endonuclease means. So the zinc finger nuclease is another endonuclease, which is a engineered protein that can, they use the zinc finger protein to target a specific DNA spot for accurate gene editing. So we have meganuclease, we have zinc finger nuclease. Then the third one is the talent. The full meaning is transcription activator like effector nucleus. Is another nucleus as well. And what is the feature of this talent? It is easier to customize than ZFN and widely used before the next technology we're going to be talking about. So the last technology we have here now is the CRISPR-Cas9 system, which is the what I call it the most popular and the most widely used genome editing because it comprises different sectors. So it uses the guide RNA to direct the DNA to cut. It is simple, it is fast, and it is versatile. That is why I said it comprises of many, many features. That is not just only limited to cutting. Do you get the point now? Are we together?
Yes, sir.
All right, thank you. Please, if you have a question, you can just call my attention to it so that we'll take it together. But if we are fine, then let's go on. So, this is more or less like what I say the evolution of the genome editing technology from the meganuclease, which is also called the homoendonuclease, to the zinc finger nuclease, to the transcription activator like effective nuclease, to the CRISPR-Cas9. This has been the evolution of the. And one thing that should come to your mind is that they were using one before, but because of the challenge or the limitation of this particular one, a new one was brought in. This new one adds its own challenge and limitation, gives room again to new one, and like that, and like that, and like that. As a matter of fact, even though the CRISPR-Cas9 is currently the one that's still being, what I say, being used the most, the scientists are already developing another one. It's just that that one is still under trial. I think it's called RNA library something. I've forgotten the name now. I will check later. It's called RLR. So, but for now, the most widely used is the CRISPR-Cas9 system because of its versatility. It is not just only meant to cut. By tomorrow, we're going to see more of what I'm trying to say because we're going to be specific about the CRISPR-Cas9 system because that is, I mean, why do we want to still be talking about zinc finger nuclease, mega nuclease when it is not being used again? However, because today is a foundational class, it is best we get ourselves familiar with all of them, not just jumping from the gene editing to the one that is being used. No, because once you understand all of them, it gives you that upper hand to design your experiments. So that is why this part is very, very crucial and necessary. So the 2011 method of the year, uh, it is ZFN and the talent. And meganuclease recognized for advanced genome editing. But by 2015, the breakthrough of the year, the CRISPR-Cas9 was honored for revolutionizing gene editing, and speeding discovery. So you can see the evolution now. As at 2020, I guess, I think the Nobel Prize winner in chemistry, Dodna, they won the Nobel Prize in chemistry because of this CRISPR-Cas9 system. That was in 2020, if I'm correct. So you can see the revolution from meganuclease, from Zetaphane to Talent, now to CRISPR. So that being said, we'll try and look at them one by one, how they work. Like I say, it is best for you to understand how they work so that when you are designing your experiment, it makes it easier for them. If you just limit your understanding to just one part, it's not going to be good for you, especially if you really want to specialize in this area in just the way I do. It is best you get yourself, would I say, familiar, and get yourself soaked in the knowledge. Okay, so we can see here, we can see the mega nucleus, we can see. Are we seeing the slide, please? Are we seeing the slide?
No,
All
sir.
right. So what's the situation? You see,
From my own
Yes,
end,
please.
what about now?
yes, sir.
Yeah, please. Whenever you don't see the slide, don't wait
I'm
until
still
I ask.
seeing genome editors.
Oh, are you sure that one is not network? Okay.
Okay.
I'm saying the one after genome editor.
The
How this
image,
mega
right?
nucleus mean? Yes, pictures.
Okay. So the person that is still speaking on medical, you can just refresh. I wait to get an on, please. Okay. All right. Thank you. So, um, like I said, please don't wait for me to ask if you can see the screen before we now, so that we won't. I won't just be talking and being an assumption. So whenever you don't see the screen, because I'm doing a slideshow, so whenever you don't see the screen anymore, just signal to me so that I can find a way to bring it back. Okay. All right. Thank you. So I'm going to on the slideshow again. Can we see the screen now? Is it visible to everyone?
Yes.
All right. So that means we can continue. Please, if you cannot see the screen, just let me know so that I don't. Remember, it's just like the word edit. They're going to make an edit, probably create a space. So anything they do has to lead with double strand break. So when there is a break, what happens? So this is points I want you to now start getting yourself, or would I say, conscious of what you're learning, because your learning starts from this point. When you use whatever, maybe you use your mega nucleus, maybe you use your zinc finger nucleus, maybe you use your talent, maybe you use your CRISPR Cas9. One thing is common: there is going to be a double strand break. And if there is a double strand break, you wouldn't want the double strand break to remain like that because you might eventually kill the organism. They ought to come back to their natural states. They ought to come back to their to the way they are before. Either you're trying to introduce a mutation, or you're trying to add, or you're trying to substitute whatever you were trying to do with a CRISPR, any of this technology. You ought to bring them back to the way they are before, which is the double strand break. So now we now have the two major repair mechanism. If there is a double strand break, it should be subjected to a repair mechanism. When you have, even in your normal biology that is going on in the cell, especially if you're studying cancer, or let me say during the cell cycle control, there sometimes be a DNA damage, and that is when your cell cannot produce p53 protein to make arrest, to code for another protein called the P21. Then all of those things happens in the cell so that the cell can be arrested and repaired. So in this case now, remember this is artificial. So when there is a double strand break, the gene should be subjected to a repair mechanism. How do they repair? So that they can come back to the way they were before. So we have two repair mechanisms in this case. The number one is the non-homology end joining. Some like they call it microhomology mediated, but the most popular one is the non-homology end joining, which is NHEJ. And we also have the homology direct repair, or you can call it homology recombination. Whichever one you choose to call it is actually fine. But for the sake of this class, let's just stick to one. So the first one will be non-homologous end joining, and the second one will be homology directed repair. So I'm just trying to explain how both of them works because it's very very important. If you don't understand this part, then whatever experiments you're trying to carry out in gene editing will be useless. Because okay, what is the point of you trying to use the gene editing to introduce whatever you want to introduce, and you are not subjecting them to repair mechanism? Even if you're using it in the plants, using it in bacteria, using it in animal, using it in cell line, they're going to die. So it is expected of you. It's just like you want to do surgery. You open the whatever, maybe the stomach and all of that. It is expected of you, whatever you are done, close it back. Because if you leave it open, the person will die. Just imagine that same analogy to what we are talking about here. So when you create a break in the DNA, in the gene, it is expected of you to bring them back together to avoid the cell being subjected to apoptosis or necrosis or whatever stuff that is going to happen that causes them to die. Now, how which you will now want to ask me which one is preferable, or let me even give the context of both of them. So I will now ask us which one is preferable. If I want to see it in the answer already. So the first one, we said we have the non-homology end joining. From the word homology, homology means something of the same substance. Homologous, homology, homologous, homology. Just see it as something of, or would I call it a twin brother or twin sister? Let me just use the layman terms now. They are the same. Do you get a point now? So non-homology means end joining. So what it is self-explanatory. It's basically saying if there is a break. Take for example, this is your sequence. Can we see the mouse? Are we together? The brother. Yeah. Can you see the DNA the DNA strand? Okay. So I'm just trying to explain something here now. So I'm trying to talk about the difference between the non-homology end joining and the homology direct repair. Like I said, both of them are the repair mechanism that are important in gene editing. If you are doing your gene editing experiments, you have to choose one between both of them, depending on your aim, depending on on the objective of your study or what you designed your study to be. So now I'm trying to explain how the non-homology end joining works. Then I will explain how the homology direct repair how it works. For the non-homology end joining, you can see the DNA fragments here. So this, can I continue? All right. So what, irrespective of whatever technology you're using, one thing that is certain is there is going to be a break. As you can see, this is the our native fragment. This is the native strand. Because of the gene editing, they broke into two. There is a break here. There is a break here. So this break, after we were done with whatever we wanted to do, additions, insertion, deletion, whatever we wanted to do, we are not going to subject it to a repair. So let's see when we subject it to non-homologous end joining, what is going to happen. So what is going to happen is that after you've deleted, probably maybe you deleted this particular region now, this particular region. So you are not going to introduce anything to them. So probably you just knock, you do a gene knockout. So when you do a gene knockout, this place will be deleted. So this separated strand will just come back and ligate together. So in this case, you are not introducing anything. This is more or less like a gene knockout. You're knocking out a particular sequence. So they are going to like it together, and this is what you can see here. This is the point of ligation. So it's going to disrupt the gene. In a case of cancer now, maybe you have a defective sequence or a mutation, and you just want to introduce that mutation, or you want to cause a mutation, delete that particular sequence. So you are not going to subject it to you are not going to introduce any sequence. So you're just going to delete that part alone. Then let me use layman terms now. Join them together, and this is non-homologous end joining. Now, for homology direct repair, it is a different case, and I think this is the most widely used because of the objective. In this case, now let's imagine again that we have a double strand break here. So we are now trying to subject it to homology direct repair. The homology means you have a template that you want to, okay, after I've created a double strand break, I've created a space here, and now I have what I want to introduce into the strand or into the sequence. Maybe the sequence I deleted here is GACT. So I now have a template. My donor, we call it donor template. It's more like you are donating that particular template to fill up the space that has been edited, to fill up the space that has been knocked out. So I'm going to have this my donor template, then subject it to this space so that they can join together. So you can see the difference between them. This one doesn't require a template. You just create a double strand break, then they come together and join. Why this one requires a template? So in this case, you can see a template here, and this is the result after the template has been introduced. So you can see the sequence here, you can see the sequence here, and you can see the template here. This one no template, this one template. Now I'm going to now ask us a question to just see if we are truly following or we understand this explanation. In these two, which one is more prone to error? In these two repair mechanism, which one is more prone to error and why? So if you want to answer, I believe Christiana should be able to help us to coordinate that. And I would actually prefer the person to just signify and talk because I want to see how we reach.
Okay, I think the homology directed repair is more prone to error because you're adding a template, and if there's a mistake, it could lead to an error. So I think the HDR is more prone to error.
Thank you. What's the name again? What's the name? All right. Thank you, Peace, for that wonderful answer. So I would need three answers, then we'll move on. So next person, please.
Yes, 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 I can hear you. Can you hear me? I think the homology end joining is the one that is prone to be a mistake, because it does not have template, and template is basically like the blueprint for something.
All right. Thank you. Are you on right?
Yes, yes, yes.
All right. Thank you for that wonderful answer as well. I love the fact that, like I said, Don't be scared of if you're wrong or right. I love when students answer irrespective of what the answer is going to be. The point of you trying to answer is at least you understand. So let's have the last person, please.
Yeah. Um, I think it's non-homologous end joining, cause the end may be trimmed or filled in correctly, something like that. Yes, the end may be trimmed, sha. So I pick non-homologous.
All right, thank you. So we have two people saying non-homologous engine is more prone to error, and we have one person saying homology direct repair is more prone to error. So I love the confidence you guys put in. All right, please go ahead, please.
Yeah, I think it is the non-homologous end joining that is prone to error because it doesn't use a template to guide the repair.
Oh, okay.
So since it doesn't use a template to guide the repair, it can cause gene mutation.
All right. Thank you, Abimbola, for the wonderful response. So I'm glad, like all of us were together in this. In this, so you've all answered very well. However, like I said, this is a place of correction and learning. So, peace. Your answer is wrong. So it is not going to be homology direct repair that is prone to error. It is going to be a non-homology end joining. Just like the reason they gave. In this case, you are not introducing anything. Imagine you taking out something and you just putting them back together because you're trying to solve a problem and you're not providing solution. That is just a simple way to explain this. You're trying to solve a problem, but you're not providing a solution. In this case, your solution is your template. So in on the bid of you trying to solve a problem, you knocking out that gene, and they come back together, it might lead, don't let me say it might lead to another genetic disorder because you don't even know. Remember your codon, you have your triplets, genetic codon. So, and this codon codes for amino acid, which produces your protein. So, take for example, you have a GAC before, or let me say you have a I think GAG, which one codes for valine again? I can't remember. But let's just say it codes for valine before, and the valine is responsible for a particular process in the cell. So if you knock it out and the GAG is now, so when you've taken out the AG, so but they will just ligate with the next available codon, which can, so it's going to disrupt the O flow. It's going to disrupt the O protein expression, which can lead to another thing. So in the bid of you trying to solve a problem, you are now creating another problem. Do we understand now? So it is much more prone to error because you're not even sure of what is going to happen next. You're not sure of the next sequence that they are going to combine again to form another protein. And that can lead to another genetic disorder or disrupt the whole gene. But in the case of homology direct repair, it's not that maybe you're still not going to have error, but that's why you use the word prone to error. Of course, there is still error. We get to see this when I go ahead. There's still error here, but it is minimal. It is controlled. Error in homology, error is in the non-homology engine is not controlled. You don't have control over it. But here, the error can still be controlled because you design your template. You design what you want to put inside. You're not just filling up with just what you don't know. You're filling up with something you've designed yourself. You know you are sure of. So your error will be minimal in this sense. So thank you guys for that beautiful response, irrespective of whatever you answer. Don't feel bad. It's good that you answer them so that you can get corrected, and it's going to help you to even remember, you know, after this time. So let's just quickly run down through. So I just try to use that to explain how the repair mechanism happens, so that whenever we now talk about meganuclease, talk about zinc finger nuclease, talk about talent, talk about CRISPR-Cas9, we all know that they are going to be subjected to the same repair mechanism, irrespective of whichever technology you're using. So that's why I took my time to really explain that part because it's very, very necessary. Now let's just get familiar with each of these technique one by one. So we have the meganucleus, just like I said. They are endonuclease and it is discovered in the late 1980s. Like I said, it's one of the first genome editing technology. So you can imagine 1980 till now, time has gone. So it recognizes and cuts a large DNA sequence up to 14 to 40 base pair. So the Liglidae family is the most common and well-known group of the meganuclease, which is named after a particular conserved amino acid sequence. The meganuclease as well, it causes less similar toxicity. Of course, we know what that means than the zinc finger nuclease due to its more stringent DNA sequence recognition. I think I should still have okay, maybe I don't have a particular picture describing the process. Maybe later we'll get to see that. And why is this? What is the problem with this particular meganucleus? Like I said before, if you discover something new today, if you use it for some while, you get to notice that oh, there's a limitation in this thing, and that gives room for innovation. That gives room for betting new things. Oh, okay, this has been associated. Even the CRISPR-Cas9 that we are even talking about today, there's already a limitation to it. That is why they are trying to now discover something that is even much more better. So science is evolving, science is moving. So the major limitation in the is high cost and then time required to engineer a sequence-specific meganucleases, unlike the Xerophane and the Talon, which uses a combinatorial method. So that is just a background of what I say the foundation of meganucleus. Then we move on to the next one, which is the zinc finger nucleus. So how is it discovered? It was
Sorry,
discovered.
I can't see this thing.
Okay. How about now?
Yes, I can see it now.
Okay. What
This
about
is
now?
mega nucleases.
Okay. So can you
It's
see
still on
zinc?
mega nucleases.
Can you see zinc finger
No,
nucleus?
not yet, but I can see mega. No, no. It's the mega nucleus I can see.
So what about others? What is other thing?
I'm still seeing the diagram. Oh.
Ha, brother, big. So is it my own network or
Is it
maybe
my
it's my
you are
I
now using
I
finger
can
or
see it now.
I can.
so what are we saying now?
Yes, sir. From my end, I'm saying zinc finger nucleuses.
Okay, is anybody still seeing an image?
no sir?
No, no.
Uh-huh. Because I've said we have left that person behind though. Okay, so let's continue. So maybe I should just stay. I shouldn't do slideshow, I guess. Let me just increase this a little bit. Okay, so the next one is the zinc finger nucleus, which we're going to talk about briefly. And I remember the first one was discovered when? Okay, let me take it back. Meganucleus was discovered when?
1980.
Okay. So this one was discovered 5 years later. So you can see what I'm saying. All because of the limitation of that one, it led to the innovation of zinc finger nucleases. So it was discovered in 1985. And what does it do? It engineered protein that specifically binds to a DNA sequence. Maybe in the next slide, we'll see how it works. So the mechanism is going to create a double strand break, just like what I've explained earlier, at the targeted DNA site for genome editing. And what is important, so it gives room for double strand break and enable precise site-specific mutation. Please, can that person mute the mic? Oh, thank you. So the cell response is going to trigger a natural DNA repair pathways, which I've explained before, the homology direct repair, which can also be called the homologous recombination, and non-homologous end joining. All depends on how you design your experiment. All depends on your objective, all depends on your goal. So that depends on whichever pathway, repair pathway or repair mechanism you want to use. So what is the outcome? It allows accurate gene deletion, insertion, modification to produce a tailored cell line. So now let's see how this guy works. Can we see the image?
Yes.
Yes, please.
Thank you. So um, this work, like I said, is an endonuclease. So there are two major parts, two major components of the zinc finger nucleus. Number one is the DNA binding domain. So please get yourself familiar with all of these things. Is the DNA binding domain, and number two is the DNA cleaving domain. First one, the DNA binding domain, and the protein which I've mentioned earlier is called the zinc finger protein. So the word finger, can we see this guy? Can we see it?
Yes.
So it looks like finger, right? More like your tongue. Can we see it? Does it look like a thumb? So that is where the name
Yes.
was gotten. Okay, thank you, thank you. That is name the name was gotten from, because of the thumb-like physical structure. That is why they call it finger. So the protein is called zinc finger protein. That is ZFP. So it's actually the protein responsible for the binding domain. And the second one is the cleaving domain. And the name of the enzyme involved in this cleaving domain is called a FOC1 enzyme. It's a restriction enzyme. Of course, we have different. I want to believe we know what restriction enzyme is, because I believe our molecular biology mentor must have probably taught us about restriction enzyme. So I don't even need to go there. So the FOC1 is a restriction enzyme, and that is what is going to be using to cleave the would I say the cell or the gene rather. So, but then how does it work? So once you understand this part, you will going to understand the remaining. Once you understand how it works, then you will understand the remaining uh, gene editing. As a matter of fact, the zinc finger nuclease is still being used today, though it is not as being used as the CRISPR-Cas9 technology. So how does it work? It's going to use the zinc finger protein, which is a DNA binding domain, to recognize three base. So please take note. The binding domain is the function is to recognize a site you want to target. That is why I said if you understand this, then you understand CRISPR-Cas technology. They are both using the same principle. So the DNA binding domain is going to help you to recognize a particular three base. As you can see here now, this particular finger is taking a GAT. This one is taking another three. This one is taking another three. This one is taking another three. So it's going to recognize three base pairs of the DNA. And once it is recognized, it's going to associate itself. Once you deliver it to the cell, it's going to associate itself with the DNA cleaving domain. And like I said, the enzyme responsible for the cleavage is the FOC1 enzyme. So once there is an association, when there is a binding between both of them, the FOC1 enzyme is going to be activated. And when there is an activation, so just give me a second, please. So when there is an activation of the FOC1 enzyme, is going to lead to cut. It's going to, of course, because the major work is to cut. So the binding domains help you to recognize the site you want to cut, while the FOC1 will be the one to do the major chords. As you can see, that's why you have something like here, like a scissors. So that's how the zinc finger work. And once it is done with its work, the next thing is to subject it to repair mechanism, which we've explained earlier. Please, are we together at this point?
Please, I have a
Yes, sir.
question.
Yes, sir.
Please ask.
I'm kind of finding it confusing, like the binding domain and the cleaving domain, and how it works with the zinc finger protein and um the other protein, the Focon nucleus. Like, what's the order of things? It's kind of confusing.
Okay. Thank you for the question. So it is not confusing. Just listen attentively. Like I said, when you are talking about the zinc finger protein, the zinc finger protein is the DNA, is the protein responsible for the binding domain. That is why I told us that we should look at the structure. It looks like a finger. What it does is for binding. That's why we call it the DNA binding domain. But the protein responsible is what we now call the zinc finger protein. Is that clear to you? First,
Yes,
so
yes.
we have the DNA binding domain, but the protein doing the work is the zinc finger protein. And as well, we have the DNA cleaving domain, their domain. But what is actually doing the cleaving is the FOC1 enzyme. So just imagine the zinc finger protein and FOC1 enzyme are more or less like or let me say the DNA binding domain and the DNA cleaving domain is the household name. But what is actually doing the work is now for the DNA binding domain is the zinc finger protein. What is doing the work for the DNA cleaving domain is the FOC1 enzyme. I wouldn't know if that is still clear enough to you.
That's correct. Thank
Are you
you.
sure? Are you sure?
Yes, yes.
All right, thank you. So, does anybody still have any question in regards to this, or we should move on? In the absence of none, I guess we should move on, right? Okay. Thank you. So that is how the zinc finger works. So once you design your DNA binding domain and you have your enzyme, then you introduce them into the cell. Then it gets activated, and whatever assignment you've given him to do is going to do it. So you just deliver it to the cell, and it's going to work. So let me move on to the next one. So you can see, I don't really need to explain all of these things. These are what I've explained before. You can see the FOC1, you can see the ZFN protein, recognize the site, then make the cut. Then after that, subject it to a repair mechanism, which I think I've explained before, the homologous recombination and the non-homologous end joining. So I really don't need to beat around with that. So let's move on to the next one. So the next technology is the talent. So how does it work? So it is similar to how zinc finger nucleus work, just a little bit modification, but a very close similarity. So it as well use in fact they are almost the same thing. It is just how the bind that makes the difference. Now, if I'm going to ask us, when I was talking about zinc finger nucleus, how many base pair does it bind to? Just need one person to give me an answer.
Three.
Okay, thank you. That means we are following. So in this case of Tali now, it's quite different. Of course, it uses restriction enzyme as well, which you can see here is still a FOC1 enzyme. They as well have a binding domain. In this case now, the binding domain is called TAL effector. Also, they have a cleavage domain, and the cleavage domain is FOC1. So we can see the difference between them. The DNA binding domain for zinc finger nucleus is zinc finger protein. In this case now, it is TAL effector, but the cleavage domain still remains the same, which is the FOC1. So how this is quite different is that instead of recognizing three base pair, they recognize a single base pair. Just a single, it recognizes a single base pair. Then once it recognizes that, associate themselves with the fork one, and then boom, it's going to cut. Please, do we understand? So that's why I said once you understand the zinc finger, then every other thing makes sense. So the only difference between the zinc finger and this one is number one, the binding domain is different, and number two, it recognizes just one single base pair instead of three base pair. Then every other thing the same. Of course, it's going to lead to double strand break, then subject to repair pathways that you want them to subject to, be it the homologous recombination or be it the non-homologous end joining, then everything still remains the same. So these are just a few, what I say, the challenges or features of the talent. The talent construct functions similarly to design zinc finger nucleus, which I've explained, and they have advantages in targeted mutagenesis. They have high binding DNA specificity compared to ZFN. They have lower off-target effects compared to ZFN again. Like I said, one problem with ZFN gives rise to innovation of talent. They have easier construction of the DNA binding domain. Of course, you can imagine recognizing one base pair to recognizing three base pair. So this one makes it easier. And as well, it offers the greatest precision due to maximum theoretical distance between the DNA binding and as well the nucleus activity. However, with all of these things now, it is still limited in its own way. And the limitation is what gives rise to the next one, which we're going to be looking at, which is the CRISPR-Cas technology. So, please, at this point, are we together now?
Yes, sir.
I hope I have not lost it. Okay, okay, thank you. So now we're going to be looking at the last technology, which is the CRISPR-Cas9 technology. So the CRISPR is an acronym, and it stands for the clustered, regularly interspaced, short palindromic repeat. Come again. Clustered, regularly interspaced, short palindromic repeat. Before I go on, can anybody say to me what palindromic repeat means? I just need like two answers. Can anyone give a try? Don't worry if you're correct. Don't worry if you're wrong. Just tell me something. Anyone?
Okay, good evening, sir.
Good evening, Ma.
Good evening, sir. Can you hear me, sir?
Loud and clear. Please continue. Loud and clear.
Okay, sir. So I think palindromic repeats, like when you read it both forward and backward, it reads the same. It has the same letters or digits.
All right. Thank you. Does anybody have any contrary opinion to that? All right. If there is none, so think what's the name, please? Okay.
Olajumoke, sir.
So Olajumoke, thank you. So that's a very wonderful to the 3', and from the 3' to the 5'. Thank you for that. So I just want us to be aware of that. Okay, so like I said, of that, say the full meaning of CRISPR is short palindromic repeat, and they are segments of the DNA of that bacteria harnessed as a form of adaptive defense against what I'm trying to say. So, how does this work? Um, Another question I will be asking us now is that what do we refer to as bacteriophage? Another person, aside from our Jumoke, another person can just try to help us with that. What is a bacteriophage?
Bacteriophages are viruses that attack bacteria.
So you can, whenever you
Infect bacteria.
can just signal whenever you're going to respond to answer so that we don't have the parents in the air. It's kind of noisy. So can you come again, please? Okay.
Is a
Virus that infect bacteria.
virus.
Thank
Infect
you, Abimbola. You can mute your mic now. Thank you. So now imagine when the virus, I'm just trying to explain how this CRISPR-Cas9 comes in place. Imagine a virus has been terrorizing the bacteria for the longest, like it has been infecting the bacteria, killing the bacteria, doing all sort of things to the bacteria. So at some point, the billion time, I have to defend myself against you. So what can I do in this sense now? Is so I have to create an adaptive immunity so that whenever you come, you won't be able to overpower me again. So I'm trying to give this storyline so that we can relate it of the CRISPR-Cas9 will be confused. So that's why I try to use a relatable analogy so that we can understand. And this is how CRISPR-Cas9 comes in. And the bacteria just come like, oh no, enough is enough. I'm not going to be bullied by you anymore. I'm not going to be subjected to your, would I say, treatments anymore. So I have to develop something that I will be using to fight you and as well build an immunity against you. So this is what it get interesting now. So this has been going on and on. So at a particular point, the bacteria now develop a mechanism so that when the viral comes in, it's going to, what I say, take in the sequence of that particular virus. Are we together now? So it's going to take a sequence of the viral DNA is going to be incorporated into the bacterial genome. It's more or less like you came to kill me now, and I took something that I can use to remember you. So it's going to incorporate the sequence of the viral DNA, incorporate it into its own genome. So it's going to serve as a memory, as a past infection. Especially take an analogy like an assassin wants to come and kill someone, maybe quote and unquote on in a lucky day, he couldn't kill that person. So the person was able to take a picture of that assassin and then store it. The picture will serve as a memory so that oh, this is the person that comes to assassinate me. So it's going to take a picture. Just imagine the picture as a viral DNA. Going to take a picture so that whenever the assassin is coming again, you can recognize the person. So you can recognize the person, and if there's a need for you to deploy your own, would I say, self-defense or whatever mechanism you want to use to counter the assassin, you can do that because you can easily recognize the assassin. So the analogy I just used is what is going on here. So the virus in this sense is the assassin. So it is going to come and want to attack the virus, then I'm sorry, attack the bacteria. The bacteria then take the sequence of the viral DNA, then bring it to its own genome, so that whenever the virus comes again, it can be able to remember. It can be able to, there's a genetic memory that is being developed for a past infection. So when the viral comes in, it's now going to use its own memory to launch attack. I don't want to sound too much complex. That's why I'm using all of these basic terms. It's going to use it to launch attack against the virus. In that case, the virus won't be able to kill the bacteria. The bacteria will use its memory, use it against him, then inhibit the virus. So when scientists now discover what is going on inside this bacteria and virus in this their relationship, what is going on. So those process that happens is what scientists now, okay, can we now artificially create something like this? That's why you'll be hearing something like the trace RNA, cRNA, guide RNA, blah blah blah. We'll get there later. So whatever that is going on, just like the same way PCR was made, or for the PCR is what is already going on in your body before your DNA replication, and the scientist, what's the name again, Caribemulis, mimicked what is going on in the body into an artificial replication. Imagine the same thing here now. So whatever is going on in the virus and the bacteria, the scientists now mimic that particular step to now see, okay, can we now discover this, maybe to use it for killing a defective gene, or taking out whatever gene that is causing the disease. So that is how the CRISPR-Cas9 is being developed. And the Cas protein, the Cas mean the CRISPR associated protein, just like you can see the CRISPR associated protein. So it's going to recognize and process the viral derived sequence, enabling the precise cutting of the matching viral DNA to neutralize the threat, which is what I just explained now. So it's going to recognize the memory, recognize the DNA sequence, then launch attack against it, then combat the virus until it dies. So by delivering the plasmid carrying the Cas9 gene alongside with a customized design CRISPR sequence into eukaryotic cell, scientists can then edit the genome at a specific location. So all of these jargons I've explained now is what the scientists will now take, design, and then this is what we are now calling the CRISPR-Cas9, the CRISPR-Cas9 here and there. Now, that is just the foundation of the CRISPR-Cas system. So how does it work? So like the ZFN, like the Talin, this one too has its own mechanism. Like I said, they are still the both, all of them are the same principle. So in work, first thing you need to understand the components of the CRISPR-Cas9. Now let me ask us before we move on. For ZFN, what are the components? What are the two components? Anybody? Can I get a response? I hope I'm not the only one on this call.
Please repeat the question.
So I said for the ZFN, what are the components? What are the two components?
A DNA binding domain and DNA cleaving domain.
Thank you. But I want to believe it is not only Abimbola that is in this class. So it will be expected of you to as well say something, okay? All right. Thank you, Abimbola. We have the DNA binding domain and the DNA cleavage domain. Okay, another person, the DNA binding domain, the DNA cleavage domain, what are the enzyme, or would I say the protein there? Another person aside from Abimbola, please. Let's make it faster. So if we don't answer, we assume that everything has been saying since are just nonsense.
The DNA binding domain for zinc finger nuclease, the protein involved is zinc finger protein, and for the DNA cleavage domain is the Focon enzyme.
All right, thank you. At least thank you for making me feel good. Okay, so at least that's a confirmation that I'm not just saying, I'm not just deceiving myself. Okay, so for the CRISPR-Cas9 system, we have components as well. And the two components we want to talk about is number one, the Cas protein. In this case, now the Cas9. Of course, we have different Cas. We have Cas9, we have Cas12, we have Cas13, we have Cas3. All those ones you can read up on your own different type of Cas. You can read them up. However, the most common used one now is the Cas9 system. The Cas9 also has its own, would I say, modification. You have we have CRISPR Cas9, and you have the normal Cas9. The difference between them is one is D. The D means you can either call it dead Cas9 or deactivated Cas9. In that case, it's not going to do anything, it's just to recognize. But let's focus on the general one, which is the normal Cas9. The Cas9 is an enzyme. The same way you have FOC1 in the zinc finger nuclease, it's the same way you have Cas9 in CRISPR-Cas9 system. The Cas9 is an enzyme and its work is to cleave. Its work majorly is to cut because it is non-specific. We now have another component which is the guide RNA. Sometimes we call it the single guide RNA. So these are the major two components of the CRISPR-Cas9 system. Now, how do they work? So when you're designing your experiment tomorrow, we're going to be designing a guide RNA. We're going to pick a gene. Then tomorrow is going to be, if you have your laptop, it will be better. So tomorrow is going to be more of hands-on. Like I said, this is not just about teaching you theory. Tomorrow we're going to do hands-on. So try as much as possible to have your system with you. If you have phone, it's still good, but preferably system. We're going to design the guide RNA for a gene. So what you're going to design in your experiment, firstly, is the guide RNA. You're not going to design your Cas9. Your Cas9, like I said, is an enzyme. It doesn't recognize anywhere. Its own is just to cut. But what is going to guide it to cut is the guide RNA. That's what they call the guide. Take what I say, create a mental picture of this. You have a blind man. You have a blind man with a cutlass. Just fling it the cutlass, and in that case, it can cut anybody. It can cut anything because it's not seen. His own is just cutting blindly. Now imagine another person that is guiding the blind man. Oh, this is where you should cut. Don't cut this place. Cut here. Don't cut here. Cut here. The relationship between the blind man and the person guiding him is what is going on in Cas9 and the guide RNA. I hope the analogy helps. The Cas9 is cutting non-specifically, but the guide RNA will now be the one to tell him, please come and cut here. Please come and cut it. The guide RNA does not cut. It's only guide. The Cas9 is not specific. It only cuts and cuts at a specific place with the help of the guide RNA. I want to believe that will make sense to us. Now, another thing you now need to take note of is something we call the PAM site. PAM. The full meaning is protospacer adjacent motif. So the PAM site, of course, you have to understand this place so that by tomorrow when we are designing our guide RNA, you're going to see everything and all this nonsense I'm saying, you're going to see it. So the PAM site is usually NGG. NGG. The N means any nucleotide. So in that case, it can be AGG, it can be GGG, it can be CGG, it can be TGG. The GG is constant, but the CGG is variable. Acid is still the same. The kind of mutation is what we call silent mutation. A silent mutation, nothing is going to happen because the protein is still the same. So that is why we need to codon optimize. And we are doing it so that the guide RNA won't be able to recognize the site in the donor template. So that is what CRISPR is all about. So like I said, you have the Cas9, you have the guide RNA. Once you deliver both of them into the cell, the guide RNA is going to guide the Cas9 to where to cut. As you can see, the scissors here, this is the PAM sequence. This is the PAM site. And this is your target sequence. This is the region you want to make a modification to. So once there is a cut here, of course, like the other ones, they're going to subject to a repair mechanism, which can be non-homologous end joining, or homology direct repair, depending on what you want to do. The most experiment I've done so far with my CRISPR-Cas9 experiment has always been homology direct repair because I'm trying to achieve something. I'm trying to do some functional genomics. So if I'm doing some functional genomics, I need homology direct repair, especially if I want to study an effect of a mutation. So I need homology direct repair because I'm going to introduce the mutation into the line, maybe the cell line or whatever line you're using. So that is how the CRISPR-Cas9 works. I just want to believe it is not too complex for us to understand. That's why I try to mix the complexity and as well the layman terms together. So, I think I have the last slide here for today. So, like I said, you can see the guide RNA form a complex with a Cas9, which is called the Cas9 guide RNA complex. Of course, that can be done through cloning. I want to believe our molecular biology teacher maybe probably explain to us molecular cloning is because you can't do a gene editing experiment without you doing cloning. Especially this is the balance now. Especially if you're using a plasmid, we all know what plasmid is. Is a vector that you want to use to deliver whatever you want to deliver into the organism. So if you're going to be making use of plasmid based, if you want to do plasmid based gene editing, we have other vector, we have the lentiviral vector, whatever vector. But if you're going to do a plasmid based CRISPR-Cas9 experiment, then the molecular cloning is your friend. Be it a restriction cloning, a ligation cloning, an infusion cloning, a Gibson assembly, or DNA assembly, all of those things are your friend. There's nothing you want to do in CRISPR-Cas system without them. So when you are doing your Cas9 and guide RNA, in this case now, you might want to use infusion cloning. Not to make it complex, let's just forget about that one for now. So the guide RNA and the Cas9 is going to form a complex, and the complex is going to be delivered into the cell. Once it is delivered into the cell, the Cas9 get activated. Then look, because of the design of your guide RNA, you have designed it to go straight to this particular site. It will then bind to the target site. Then, because of the palm site, will be able to recognize it, and then the Cas9 start cutting. After cutting, then leads to a repair mechanism, like we have explained, either the non-homologous end joining or the homologous direct repair, depending on your experiment. So this is how the CRISPR-Cas9 system works. For the D-Cas9 that I made mention earlier, it doesn't cut. It doesn't cut. It is mostly used just like the way real-time PCR works. It's used for detection. When you have your GFP and the D-Cas9 together, especially in the vector like your D-Cas9 KRAT vector. So it's just going to help you if you want to study, would I say you want to do a conditional knockdown. It is not going to cut. It's just probably either repress the gene is still there, but it's not as expressed as before, or just help you to signal, just like the way your real-time PCR will work, because the Cas9 is deactivated, it is dead. But in this case, in normal Cas9, it is going to cut. So that's just the difference between them. We also have other Cas like Cas13. I think Cas13 also work like D-Cas9. It's majorly for diagnosis. It's majorly for detection, not for functional genomics assay. So with all the nonsense I've been saying since, that is how the gene editing, that is the foundation of our gene editing class. By tomorrow, we're going to get ourselves, of course, familiar with some terms, which is here, before we proceed to our hands-on experience, which is designing your guide RNA. I think I've done with today's class. Thank you, everyone. So, like I said, it's the foundational class for gene editing, and we try to look at what the gene editing is, which I said is used to make an edit either by adding to the sequence or by inserting, by deleting, by substituting a particular DNA sequence, maybe in case you want to do single nucleotide variants. So I said that is what the gene editing is all about. And I said it's quite different from the traditional genetic engineering, which does their own just randomly without being specific. So the specificity of the gene editing is what makes it to be preferable over genome editing, over genetic engineering. And we're trying to look at a repair mechanism of how whatever gene editing experiments you're trying to do, irrespective of the technology you're trying to use, we're trying to look at how the repair mechanism works. And we agree that we have just two major repair pathways, which is the non-homologous end joining and the homology direct repair. And I try to explain the difference between them. That a non-homologous end joining, it makes you are not using a template for your repair. It means when you delete a particular sequence, the remaining part will just come together and ligate together, which I say it is prone to error because you are not sure of what is going to happen. It's going to lead to the disruption of the gene because when this thing works in genetic code and it works in triplets. So if you have GACG and you delete C, it's going to lead to GAG, which is going to code for another entire protein and it's going to affect the whole gene sequence. So it means everything will be disrupted. And as I said, in the bid of you trying to solve a problem, you are creating more problems. And I made mention of the homology direct repair, which I said, even though it's still at its own error, but the error is minimal, which I said you're going to be making use of a donor template you design. It's just more or less like you're trying to delete something and you say, okay, when I delete this thing, I want this thing to be inserted, which is still quite better. Or at least you know what you're trying to do, especially if you want to do functional genomics now. You know what you want to introduce and makes your works much more better. It requires a template and it is not as prone to error as non-homologous end joining. And we look at the different technologies in genome editing. We look at the mega nuclease, which I said was discovered in 1980s, and it has its own limitation, which is it's very complicated to design, which makes it resorted to limited use. We look at the zinc finger nuclease, which I said it came after the mega nuclease, which was in 1985, and it has two major domains, which is the DNA cleavage domain and the clear DNA binding domain. And I said for the DNA binding domain, the protein responsible for that is the zinc finger protein. And for the DNA cleavage domain, the enzyme responsible for it is the fork1 enzyme. And I said the DNA binding domain recognizes three base pair and the D in the gene. So when there is a recognition of the three base pair, it leads to the activation of the fork1 enzyme. Then there's going to be a double strand break, which is then going to subject to repair pathways, either the first one I've mentioned or the second one, which is the homology direct repair. And we talked about the talent, which is kind of similar to zinc finger nuclease, but the difference is that it recognizes just one single base pair, unlike the zinc finger nuclease that recognizes three. And the DNA binding domain in TALEN is different. It is TAL effector, unlike the zinc finger protein in zinc finger nuclease. But the DNA cleavage domain is still the same, which is the fork1 enzyme, and it's to work in the same way, make it decut and subject to a repair mechanism. And lastly, we talked about the CRISPR-Cas9, which is the most widely used and the common at the moment, which I said it's gotten from a bacteria. When the virus comes to attack the bacteria, it develops an adaptive immunity against the bacteria by recognizing the viral DNA, integrating into it the bacterial genome, then using it against the virus whenever the virus comes to come and attack the next time. And I said the scientists mimic this process and turn it to a gene editing tools by in this case now we now have two components of the CRISPR-Cas9 system. which I said number one is the Cas9 protein. The Cas9 is an enzyme that cuts, just like the FOC1 enzyme. The guide RNA is just more or less like your binding domain in this sense now. The guide RNA, which I said is going to include a palm site, and I said the palm site is called the protospacer adjacent motif, and the palm site is NGG. The N is any nucleotide. It can be GGG, it can be TGG, it can be CGG, it can be AGG. So altogether with the guide RNA will be 23 nucleotide. The guide RNA is 20, then the palm site 3, making it 23 nucleotide. And I said when you are designing, the most important part is your palm site, because that is where it's going to recognize your target site. And I said when you design your guide RNA, when you deliver them, you're going to do a cloning, which I mentioned that molecular cloning is very, very important in gene editing experiments. Then when you do your cloning, then they infuse together and then you deliver them into the cell. So when you deliver them into the cell, you're going to, and I may mention before delivering them, that you're going to do codon optimization, especially if you're designing if you're designing a construct for your template. When you're designing your construct for your templates, you're going to modify the sequence. You're going to optimize the sequence in such a way that you're introducing a silent mutation. And I use an example. If a GAG is coded for valine, I can codon optimize it to be GCG. It is still the same valine, even though the nucleotide changes. So that is a silent mutation. The purpose is so that the guide RNA won't be able to recognize it when it gets to the donor template, because if you don't do codon optimization, the guide RNA will still after it has cut your normal sequence, it is still going to cut the DNA template as well. So that is why it is important for you to do a codon optimization. And you deliver them into the cell, the guide RNA makes a break, cut whatever it needs to cut, subject it to the repair, because of course you have transfected the what's it called now? You've transfected both of them with the donor templates and with the construct, the plasmid. So it's going to repair itself with the template, then you're good to go. So I think that is all the summary of what I've talked so far.
Thank you so
Thank you,
much, sir.
sir. Thank you
you.
very
Thank
much.
you,
Thank you,
sir.
boss.